All right, students, welcome to the second part of our week three topic where we were counting some macros, building some macromolecules. And just as a recap, kind of what we talked about in the previous video, we broke this topic into two videos where we talked about, in the first video, macromolecule synthesis and how that pertained to carbohydrates and lipids. Okay? Now, the thing I want you to remember is macromolecule synthesis is nothing more than monomers joined together to become polymers. Okay? Um, and the resulting equation with that, so monomers become polymers, and a little bit of H2O is generated. One molecule of H2O is generated for every two monomers that are put together. Okay? And this is something called dehydration synthesis. Um, and the reverse of that, if you go from polymers to monomers, you have hydrolysis. Okay. And I broke those words down to you, or broke the word hydrolysis down in the previous video. So remember that. But how this pertained to carbohydrates, you had sugars that became carbs, or like a monosaccharide to a polysaccharide. You had fatty acids that became fats or lipids, depending on how you want to look at it. And we're going to do the same thing for proteins and the same thing for nucleic acids. Now, as far as enzymes go, we'll talk about it, but enzymes are just proteins. Okay? Enzymes are in fact just proteins, and we'll talk a little bit about them, but we really talk about them when we talk about metabolic pathways later in the semester, or metabolism, depending on how you want to look at it, but metabolic pathways. Okay? So moving right along, that's the wrong slide. Let's go straight into proteins. So proteins are, like I said, if we look at like carbs and fats, those have like a negative connotation in today's society, in social society. They shouldn't, but they do. Proteins, however, are good. Proteins, people love protein. Um, so we talk about high protein diets, but proteins are the most abundant organic molecules. Organic just simply means they have carbon attached to them, or they have carbon in the molecule itself. But they're the most abundant organic molecule in living systems, and they have a variety of functions. They have structural properties, regulatory properties. They can be found in muscles, protective tissue, transport. Hemoglobin is probably the best example of this, which is basically your red blood cells, which carry oxygen around your body. Storage and enzymes. Enzymes. The enzyme I always use is one that's lactase. It's the one I remember the most. Something like ATPase is another one. But each cell contains thousands of different types of proteins, and they're built of these linear sequences. Now, if um, protein is the polymer, then these things called amino acids are the monomer. Okay? So proteins are made of amino acids. Now, there's only 20 amino acids out there, but through a combination, through varying lengths, through varying orders, we get every single protein that we can possibly get based on these 20 amino acids. It's remarkable diversity here. And it is really, really, really cool what we're able to do, or what our bodies are able to do with what seems to be such little data. So, enzymes. So, we're talking about here's the brief talk of enzymes. Enzymes, again, are just a different type of protein. this means that again, when we talk about proteins and we talk about enzymes, we're talking about proteins, but not all proteins are enzymes. Um, but basically, what enzymes are is they are just biological catalysts. They make reactions happen. Okay? They make reactions happen. So enzymes can make certain digestions happen. Here's an example. I talked about lactase earlier. Lactase is an enzyme that breaks down the sugar lactose. You may see where I'm going with this. If a person does not have lactase, this person is what we call lactose intolerant. Okay? So this is an example of an enzyme that your body, you don't necessarily need to survive. There are enzymes that if we don't have, we die. ATPase, what I talked about earlier, is one you need. If you don't have it, you're going to be in a world of hurt. But mutations to enzymes can cause a lot of damage. This is a sort of a benign one. But essentially what enzymes do is it lowers the cost of energy. And what does that mean? So an energy may require, let's just make up a number. Like let's just say it requires 10 units of energy to work. Okay. Well, what that enzyme can do is it can lower the cost to say 4 units. Still, there's some energy that needs to go in there, but that enzyme makes this possible, much more efficient. We'll get more into that in subsequent weeks. But what I want to start off with right now is amino acids. And remember what I said, amino acid is the monomer tied to the protein, that's the building block of proteins. So I'm going to go to our trusty blackboard here, and I'm going to break down this thing called an amino acid. So we're going to go to amino acid right here, which again is a subsection or the subunit of proteins. Okay. So when we're looking at an amino acid or a protein, we're looking at three separate parts. Okay. You're looking at a nitrogen portion and then two carbon portions. This nitrogen portion, uh, let me get rid of that because that's going to bother me. There we go. This nitrogen portion, you have little hydrogen and nitrogen bonded there. And then on this outside carbon, you have it double bonded to an oxygen and then an OH group up here. And then this middle carbon has an H, a hydrogen, and then a side chain that we're going to label as R. So that side chain can be a hydrocarbon backbone of some kind that's attached to that carbon. So what you're looking at here is if I take my blue here, what you have here is right here, this guy is known as the amino group. Okay? And this guy over here is known as the carboxyl group, but that's just a short name. It's a carboxyl acid. So combine these together, you get amino acid. Okay? The more you know, right? Now, in the middle here, you have this carbon, the middle carbon that we just call the alpha carbon. Okay? And this alpha carbon's bound to a hydrogen and then some side chain out on the edge like that. Now, that side chain can be anything. I'm going to give you an example. I'll give you two examples. They're probably the smallest examples. But let's take something like glycine. Okay. Glycine, again, you have the N group, the alpha carbon, and the carboxyl carbon. Okay, you have it bound to the I don't want to do this here. You have it bound to the oh, sorry, the H2. Losing my mind here. The boy, losing my train of thought here. Oh, there we go. Yeah, you have it bound to the OH group up here. Boy. And then the O group down here. And whenever you're looking at this, you look at the H up here, right? Um, Glycine, though, with respect to that other H that's up here, its other R group, this guy right here, is just another H. Okay? So H right there is that what makes glycine. It has that amino group, that amino backbone, and then it's just bounded that R group's in H. Now, if I take something like, um, let's go to something like alanine. Okay, same thing. We'll just do this H2N to the carbon to the carbon. You have the H group up here, OH, double bonded to the O. And the R group down here with alanine is just CH3. Okay, right here. All that guy, that's the R group. That's the R group there. Okay. Those R groups are what make the amino acids different. Now, they can get long, something like arginine is really long, histidine, very complex. Some of them have rings like tyrosine, phenylalanine, tryptophan, that's a really big one. But there's 20 of these. All of them have the same structural units of this amino group, the carboxylic acid group, or carboxyl acid group, and then this R subunit. Okay? So that's what we're looking at when we look at amino acids. So what does this mean structurally? All right. So when you make amino acids, or when you have amino acids, and you're trying to build proteins, again, proteins are just a bunch of amino acids put together. What does that look like? Okay. So I'm going to draw this again, switch to my green color here. So let's say you have something like H2N. You go C, you go C. I'm going to reverse this. I'm going to put the O up here. I'm going to put the OH down here, which means my hydrogen goes here and my R group up there. Okay. And then I'm going to get another color. Let's go with this dark yellow here. Okay. And then I'm going to do the same thing. I'm going to go N, and I'm going to draw this a little differently. I'm going to put my H up here and my H up here. And then I'm going to bind it to two carbons. There's my R group there, my H there. Okay. So I'm going to add these guys together. Okay. Now, when I add them together, I want you to notice something. What is an amino acid in relation to a protein? It is a monomer, and we are becoming a polymer, which means we should generate what? H2O. Somewhere in this mechanism, we should generate H2O. Well, I'll take some ink here. I have an OH group right here. I have an H here. What ends up happening is these guys fall out of the equation. H2O is generated over here. And what we're left with is if we kind of just draw a line here, H2O falls out, okay? Those guys are gone from the equation. What you have here, and I'm going to take these in the colors that we started with. And I'll be over here. Well, let me see. I'll get my other amino acid up here. There we go. Here we go. And then finally, it's double bound to the O and then the OH. Okay. And so we're not showing favoritism in the colors. What ends up happening is these guys are now bonded together. So water fell out of the equation. This subunit right here, this amino acid, bound to this amino acid. And what you have here, after the water's fallen out, is something called a peptide bond. Okay? This is a peptide bond. Um, Many of these put together, right over here, is a polypeptide bond. You may have heard of these terms, but a protein is essentially a polypeptide. Food that's just something I want you to think about. That's your food is polypeptides. That's really what it is. Um, so as more amino acids grow to this chain, it's a polypeptide, okay? Um, what you're looking for here is you have these two ends here, and this is some more nomenclature. You have this guy and you have this guy. Okay. Where the amino group is, we call that the N terminus. That's the nitrogenous end, and the other end is called the C terminus. Okay. So the N terminus and the C terminus, that's how they link up, but the ends still have some they have a directionality to them, okay? So we label one end to be the N terminus, one to be the C terminus to just kind of show where we are relative where we are to the chain. We're four amino acids in front of the C terminus. We're four amino acids in front of the N terminus, so on and so forth. Um, so what does that mean for protein structure? So again, we talked about the amino acid proteins, how they bind together, um, the peptide bond. So what does that mean for actual proteins? Okay. Well, proteins, like I said, are made up of amino acids, a bunch of amino acids. It could be a few to several. I could even begin to tell you how long some are, maybe hundreds of amino acids long. But that just means that we have to understand what they look like. So when a protein is made, and again, we're going to kind of break this down as easily as possible. You have four things to look at. One, two, three, four. And the first thing that you're going to look at, the first level structure that comes to protein is the amino acid sequence. Okay? That is very important. The amino acid sequence is the most important thing when it comes to determining what the function of a protein is going to be. Okay? So if I'm looking at amino acid sequences, just sort of this order of what they're doing. These just pretend these are amino acids. Okay. Let's just say, well, we'll start with methionine, and you have alanine, and you have isoleucine, and you have leucine. That's a good one, and then lysine, and then you have I'm trying to think of one off the top of my head, tryptophan. There we go. Um, this order tells me everything I need to know about uh, what amino acid I'm working with. That tells you what you're working with, just that identity. But the shape of it is something even more. So the first thing we look at is we look at the amino acid sequence to determine what's happening there. Um, and we also, whenever we're ordering this, so like methionine here is on the N terminus end. Tryptophan here is on the C terminus end because remember we had that. H2N on this side, and we have that carboxylic acid over here. So kind of moving in that direction. That's what we're looking at. So that's the amino acid sequence. Okay. Um, and well, I'll talk about like mutations in a minute, but the order is very important, and I'll show you why. Um, the second thing we look at, so when we look at that amino acid sequence, the next thing, what we call here on the amino acid sequence, and I'll just go ahead and say it now. Um, this is something called the primary structure. The first thing you're looking at is the amino acid sequence. Okay. Now, if I look at the next thing that determines it, um, and I'll go ahead and just write it so we see. It's going to be this secondary structure. Okay. So the secondary structure is important here because what you end up seeing is that some of these groups, and if I'll get another color here, like let's go white. So let's pretend, remember how when we go back to um, this N-terminus end and this carboxylic acid end? Well, just by orientation, just by the way that, when you're looking at H2N up here, that N kind of pulls some of the electrons over here and makes it slightly negative, and slightly negative, and H, the hydrogen, is slightly positive, kind of like with water, had that electronegativity to it. The same kind of thing happens um, over here with this oxygen, is it sort of pulls electrons this way. Oxygen's a little greedy. So what you end up seeing is you end up seeing that this hydrogen in one group tends to want to interact or be pulled into close proximity with the oxygen on that carbonyl, that group, that C terminus end. Okay? So what you can end up having is maybe, and I'm just kind of postulating this, so let's go with our amino acids. We're not going to label them this time, but it's possible that the N terminus on this group is slightly attracted to the C terminus on that group, and this one to this one. And there could be orientations along those lines. Now, what's important is if these guys are close together, like they are here, you'll start to see stuff kink up, like they start to bend a little bit. All right? That attraction varies between amino acids. So the order tells us one thing, but if certain amino acids are in close proximity, they can start to form what we call alpha helices. Okay? If there aren't a lot of attractions, then you don't have a helix. You have these straight sheets called beta sheets. Okay? So the orientation of the original or the amino acids then lead us to the interactions of the terminus groups, the C terminus and um, the hydrogen on the N terminus and the oxygen on the C terminus. Um, that leads us to have hydrogen bonds that result in either alpha helices or beta sheets. Okay, so that's the secondary structure. The next thing we look at is the tertiary structure. Okay, and the tertiary structure um, is simply, if we go back, let's just go all the way back. It's simply the reaction of your R groups. So when all of these, and again, let me get some blue down here. Let's get a different color. Let's get red, just so we can make sure we're not getting confused here. So we'll go red, and I'll move that 4 here in a second. You know, we have maybe some helix action going on here. Well, then within these, the R groups start to react to each other. Those R side chains that we talked about. And maybe this one pulls over here, this one wants to pull over here. And what you end up getting, and let me erase that 4 so you're not confused. What you end up getting, I'll put 4 down here. What you end up getting is here, and I'll draw this over here. What you end up getting is you started getting this sort of like spaghetti-like feature, and that is essentially a protein. And it's determined by the amino acid sequence, whether it's an alpha helix or a beta sheet, depending on how the carboxyl oxygen and the nitrogenous or the nitrogen bound hydrogen interact with each other through hydrogen bonding. And then furthermore, the tertiary structure is determined by the R side chains in each amino acid. So you have all that. And then the last thing that you want to think about is you want to think about the fourth thing, which is a little easier, but it's the quaternary structure. Okay? So the quaternary structure is essentially if this happens one time, maybe it'll happen about 3 or 4 times. And some more of this. And one more color. What color haven't we used? Let's use some purple. We haven't used purple. And what you end up having is this sort of 4 different proteins bound together to make a bigger protein. And those are the 4 roles or the 4 structural subunits of any protein. You have the primary amino acid sequence, secondary, the helix or the beta sheets, tertiary, the R group interactions, and then multiple subunits coming together. Okay? So what that means for us is if I were to actually show you, so if you go to this website, I'm going to drag this over so you can see. Um, you see this website. It's called Protopedia. In fact, let me just kind of drag this over here so you can kind of see. If you want to go see what proteins look like, you can see this. This is an example of a protein. This is, I believe this is like a hemoglobin protein. Um, and it's bound to some AMP in here. So it's not hemoglobin, I'm sorry. Something else. But anyway, this is just an example of protein. You see the helices acting in here, and you see that it has a specific binding site for this particular AMP. Now, what does this mean? Now, if I look at, I'll get rid of this because we don't need it anymore. Um, if you look at this from a standpoint of structure, if I were to just even go up here and let's say at the very beginning, go to my amino acid sequence, alanine I removed. Well, alanine may have been interacting with leucine, but now if it's tryptophan, if you can see that tryptophan, now it no longer interacts with leucine. And I'm making this up. I don't know specifically. But what ends up happening is that structure is destroyed, the amino acid structure. The secondary structure is different, so it could change from a slightly different alpha helix. Now the tertiary structure is down, and then finally it interacts with other subunits differently. So one mutation can have dire effects for the entire system. So, what does this mean? So you actually end up, um, where am I going here? So yeah, you have amino acids as the building blocks. Um, but yeah, each amino acid is built is bound to that amino group and a carboxyl group and the R group side chain. Okay, we talked about that. The links, we talked about dehydration synthesis, how a water is formed by the dehydration, but that comes between 2 peptide bonds. Um, and here's just a better representation of that. Amino acid 1 to amino acid 2, a peptide bond forms, H2O released. The 4 levels of protein structure. Primary structure is the sequence of the amino acids. Okay. The secondary structure relates specifically to within the chain, the N terminus and C terminus, more specifically, the hydrogen here, the oxygen here, hydrogen bonds. affect whether it's kind of coiling or sheets of that nature. Okay? That's the secondary structure. The tertiary structure deals specifically with the R groups. Once they're in these forms, maybe some R groups are sticking out right here. And these, through ionic bonds, hydrogen bonds, some covalent bonds, contort the protein even more so that you get this sort of spaghetti monster like this. And then if I link four spaghetti monsters up, I get the quaternary structure. Okay, so what I mentioned earlier is if I change one amino acid, if I'm going to take this to the extremes of just talking about, say, just one amino acid difference, um, sickle cell anemia is probably the biggest thing I can think of because all it does is it changes one amino acid. So there's a mutation. It's basically valine is replaces glutamic acid. And it's just this, and it's the sixth amino acid in the chain. We've actually sectioned that to the point where we know exactly what gets changed and what. So it's a beta chain of hemoglobin, but just that causes a healthy red blood cell like this to look like that. Well, what that does is it limits O2 from going through the body. The red blood cell can't carry oxygen throughout the body. It's a minor change, but it has devastating effects. And again, it's because once I change the sequence, I then change the interactions of the hydrogen bonding in the secondary structure and the R chains in the tertiary structure, and subsequently whether it can bind to other protein subunits in the quaternary structure. Okay, so that's amino acids and proteins in a nutshell. All right, so the last thing that we're going to talk about is nucleic acids. And I'm going to go back to my boards up here. And I'm going to erase, erase, we're going to erase, okay? So we're now going to go up here. We're going to look at nucleic. Ah, it's too dim. I'm not going to do that. Erase all ink. All right. We are going to go up here and we're going to do something you can see a little bit better. There we go. Nucleic acids. Okay. And when we talk about nucleic acids, we're talking about the big ones: DNA, RNA. Okay. DNA and RNA. Or if I'm going to write that out, deoxyribonucleic acid. Okay. And RNA is ribonucleic acid. Okay. Now, why is this important? Why do well, you get that DNA is important, I think, because that's where your genetic code is. That's where your genetics lie. It's what makes your features you. It's the essentially the code that makes you you. But what are we talking about when we talk about DNA and RNA? Okay. So when we're looking at like nucleic acids, we're looking at DNA and RNA. These are what we call nucleic acids, and those are the monomers. Okay. Now the polymers, I'm sorry. Now the individual monomers of these are these things called nucleotides. Okay. And there are five nucleotides technically, but there's only four in each nucleic acid. And I'm just going to label them. You have A, T, G, and C. You have A, U, G, and C here. So A equals adenine, T equals thymine, C equals cytosine, G equals guanine, and U equals uracil. Okay. Those are what comprise DNA. Okay. Now, adenine, cytosine, and guanine are in both. Okay. However, thymine is only in DNA and uracil is only in RNA. Okay. So if I make a chain of these DNA and RNA, you can tell the difference by the presence of uracil or thymine. Okay. So these are nucleotides that make up DNA and RNA. Okay. So what is DNA and RNA? So leaving this as it is, I want to break this word down for you. First, both of these actually, you see you have nucleic acid for starters. So they're a nucleotide. They have an acidic group to it. But you have here in this word, you have the word ribo. So what does that mean? Well, ribo just means it's a ribose. Okay. Which, if you see that OSE, what does that mean? It's a sugar. Okay. Ribose is a sugar. More specifically, it is a 5-carbon sugar. Okay. Now, this is actually really cool. So I'm going to explain the difference to it. So you look at this 5-carbon sugar in ribonucleic acid, and then you have this extra prefix up here, deoxy. So if you really break that down, we go to deoxy. It is D, which means removal or less or absence. And then you have oxy, which is oxygen. And so really what you're seeing is you're seeing that there is less oxygen in DNA than there is in RNA. And so if I were to break this down, if I were to look at um, these sugars, now remember they're a pentose sugar. Do I want to go to another slide? Yeah, I'll go to another slide here. Give me some room. Give me some light green here. Okay. So remember, this is a DNA versus RNA slide. Okay. DNA versus RNA. And what you have here is you have this 5-carbon sugar. Okay. And we have oxygen right here. That's for starters. We have oxygen. Okay. Now I'm going to draw this out. So right here again, and I'll just draw it out and we'll show you how this works. Okay, so you have an OH group here, an H group here. I'm going to skip this carbon right here, and I'm going to go to this carbon. H here, OH here. Come over here. You have an H here, and this is bound to a HOCH2. Okay. And now what you're looking at here is you're looking at um, a ribose, and let me go in here. And this carbon right here is the 1. Here's the 2. Here's the 3. Here's the 4. Here's the 5. It's a 5-carbon sugar in a ring-like form. Okay, so we have that. Now the difference is here, and I'll draw this in another color. Let's go some yellow. What you have here, again, this is where it's different. So in this 2-carbon right here, both are the same in that they have this H up here. However, RNA has an OH group down here. Okay? RNA has that OH group down there. This was what makes it RNA. However, in DNA, this O group is not there. And instead, you just have an H. So I'll bring this down so that you can see that there's an H right there in this case. And that is indicative of DNA. On the 2-carbon in this ring, 1, 2, 3, 4, 5, 6, or 5, sorry, on the 2 carbon right here. RNA has an OH group. DNA has an H group. Okay? That is what makes them different. And whenever you look at this, whenever you look at DNA and RNA, that is the backbone to what makes them different. Um, and now, just a little bit more clarity whenever you're looking at this. So let me draw this a little bit. So just to avoid drawing this over again, I'm going to draw. So you have your ribose, okay? On your ribose, you have 2 ends. So we're going to say this is the 1 end, and then we'll say that this is the 5 end here, okay? So on the 1 end, you actually have something that is sticking out of here, and this is called a nitrogenous base. And on the 5 end, you have a phosphate group. I think it's 2 minus. So you have a phosphate group and a nitrogenous base that kind of stick out here. And so here's your nitrogenous base. Here's your phosphate group. The nitrogenous base acts like that R group with amino acids, and this is what makes thiamine, guanine, cytosine, uracil, that nitrogenous base that sticks out the end of it, okay? So you're looking at a phosphate group and a nitrogenous base. Um, and that's what we're looking at there. So this nitrogenous base, again, it's the telltale sign. It tells us what is DNA or what nucleotide we're working with. And subsequently, it forms our DNA. So whenever you're looking at this, when you're looking at, I'm going to draw my DNA right here. Okay. See if I can still draw this the way I used to. Okay. What you're looking at here, okay, you're looking at a DNA, which DNA has this thing called a double helix. And it has 2 anti-parallel strands. It just means they move in opposite directions. This one goes this way, this one goes that way. Okay? Now, in between each strand, on each strand are the nucleotides. Okay? So on one strand, let's say you have C, G, A, T, A, C, G. That would be this strand right here, okay? Now they're complementary. So if you have it on that end, and again, we'll draw this in green going this way, the C would be bound to a G, the G would be bound to a C, sorry, a C, A would be bound to a T, T bound to an A to a T, C to G, and G to C. Now you could just use the abbreviation C to G. You can say, or you can say cytosine to guanine, guanine to cytosine, adenine to thymine, so on and so forth. Now, if this were RNA, RNA again, so if this were actually RNA, everything that is a T would actually be a U. That's the only difference. Okay, so we're looking at these bonds in such a way that they're complementary. C bonds to G. Now, the difference between C and G and A and T is C and G have 3 hydrogen bonds. A and T only have 2. Okay, so why is this important? It's because a double bond is much easier to break. So a lot of times when we're looking to start replication and we split the strands up, we split them up by going this way and this way, finding a bunch of A's and T's, or we call those like a ta-ta box, that's where we can start replication. And we'll talk about Tata boxes and replication forks later, but that just gives you a preview. So yeah, DNA has a double helix structure. Um, and your book goes a little bit further into how ribosomes create or use DNA to code and ribosomes to code for proteins. We're not going to talk about that right now. But what you need to know here, let me see. Let's get rid of this. What you need to know on at this juncture. So if I just go here, we'll just do a little recap of this section. Um, you got to know your proteins. You got to know your amino acids. Okay. And you got to know your nucleic acids. What are the nucleic acids specifically? Different nucleotides. So something like A, T, G, C, those are your nucleotides. Okay. Within that, within proteins, peptide bonds, maybe something like the alpha carbon. What is that? What is the old alpha carbon? You can go back in your notes and look, but nucleic acids to nucleotides. Um, Dehydration synthesis. It does show up in nucleic acids too, but for a sake of time, I did not include that. Okay. But we saw it in proteins. That peptide bond um, creates a water or creates a water molecule. Nucleic acids, ATCG, DNA versus RNA. What does that two carbon have to do with that? Remember, there's oxygen involved there too. Okay. We look at proteins, peptide bonds, the alpha carbon. What about the structure levels? Know all that. A question I could ask is something along the lines of, let's just say I could ask something simple like alpha helices refer to which structure? Secondary. Or I could ask you to write a little 2 to 3 sentences, or if I change one amino acid, explain to me how that affects every single level of the structures. How do the structures change at that point? Okay. So take all that, remember all that, but that's the recap. Go over these notes, read your reading. Your reading is a little bit more in depth. I'm trying to filter this out so you understand what we're talking about a little bit clearer. But this stuff again is going to keep building. So keep striving along, keep chugging along. You're going to do fine. That being said, that is all I have for this week. Um, yeah, and if you have any questions about this stuff, you can always email me. Be as thorough as possible with your emails. Just don't ask generic questions like explain it better. Tell me what you're not understanding, and I'll gladly respond to any emails you send. As always, what I always say, I like to say is stay curious. And this is me signing off. Okay. Have a great time, guys. And again, message me if you have questions. Adios.